Hello, I am Amanda Wacker.
Hola, I am Lennice Castro.
สวัสดีค่ะ I'm Asama Lekpua.
And we are the Triplicates. Welcome to the second season of the Triplicates podcast. We are three first-gen women of color completing the second year of our PhD program at the University of California, San Diego.
Last episode, we talked about the new things that second year brought into our life and what has continued from first year. But today,
We'll be talking about some spooky science stories since Halloween is coming up.
Halloween!
That's scary.
Yes, I definitely have some lab horror stories, but does anyone want to go first?
Do these have to be like our own or like from what we've heard?
Okay.
And can we name institutions?
Oh, I don't know. Let's nothing illegal, maybe.
Yeah,
Um,
something vague maybe.
Yeah. I was thinking like
nothing illegal?
things that I have done that is like, oh, what a horrible thing to have happen to you while in lab.
For legal reasons, we've done none of these. I don't know.
I just like used the wrong buffer.
Oh.
You know, like that kind of stuff,
Okay.
you know? Not
Not bad.
like
Can't
I
incriminate
stole
yourself.
anything.
Well, do you get me? Like, what if our EHS officer watches this and he's like,
Oh,
excuse
we
me?
don't have to say which lab we're in.
I agree.
I've been in
I would
like
be
7 labs
flattered
from
if
now.
he watches because
Oh.
I feel like he doesn't like any of us.
Oh
Hold on.
no. Okay. Oh, okay, go
Oh.
ahead. You can take it.
Oh my god, that's Christopher.
Hello?
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 Tell them we're recording. Okay.
Okay, well, back on track. Amanda, since
Yeah.
you are so excited and there's
Oh.
many things that
Oh.
I see you want to talk about,
well,
you should go
I
first.
picked 3. I picked 3.
You can do one.
Okay. Yeah. So I think one of the I think this is more a horror story for the grad student that I worked for at the time. But she had some like samples that she had done a protein curve to see like what the concentration was. And then she thinks that maybe one of the samples were like arranged in the wrong order, or like on the like line 7 maybe wasn't actually what she thought line 7 was. So she was like, here are the proteins. If you just rerun the proteins and write it down again, we can match like the protein concentrations from your thing to my thing and then like confirm what the protein concentration was. And I was like, yeah. And, you know, I was a second year undergrad and I was like, I know that freeze thawing samples is bad for the samples.
For sure.
So I did not let it thaw all the way. I just let it thaw like a little bit and then took some liquid off the top and then put it in the little protein curve. None of those numbers were correct.
Okay, so you didn't put the icicles in the assay. Is that
No, I did not put the icicles in the assay,
okay?
but like
that would have
didn't-
been hilarious.
No. Oh my
So
gosh.
Let me take the top layer of this.
Yeah, I was like
liquid,
probably
and
just water.
It's going to barely like represent the entire population.
Yeah. So
You live and you learn.
yeah, that I don't think my grad student had a good day that day. I'm so sorry.
no.
But did she graduate?
She did. She got out of there.
Did she need it? Was it like an essential experiment?
No, it's just like a protein curve. Like we did a lot of westerns, so it's like just to know how much protein to put in the like that kind of thing.
Oh,
It wasn't serious, but
wow.
like it was a silly thing for me to do.
It's okay.
That's why you need to clearly tell people what to do and
Yeah,
not
you learn.
assume that they know what they're doing.
It's true. Never assume.
Yeah. Anybody
Lennice, you have some?
popcorn?
Okay, I can go. I guess like I was so we knew we were gonna do this episode since a little while ago. And I was thinking, I was like, I have horror stories maybe, but I can't like remember any of them. But you know how people tell like ghost stories
Yeah.
to like warn you to
Yeah,
not
yeah.
do things
Yeah.
in order to avoid these like
Yes.
silly little mistakes. I feel like I have the equivalent to like for science, right?
Yeah.
Like
yeah.
I am deathly afraid of the autoclave.
Oh.
Or like
You think it's
even
gonna
big
explode?
centrifuges. Yeah,
Oh.
because one time I was at an internship and she was like, yeah, you have to be really careful or you can like blow half the building up.
Oh
and I'm
my
like,
They're basically big pressure cookers.
I mean, exactly. So I guess like if things go wrong,
yeah.
it can. But so that's why when I came here and I had to use the autoclave for the first time by myself, I was like, oh my God, I'm going to blow up the whole building if I don't do everything correct.
Yeah.
So I was so scared to use it, which it's fair. Like the autoclaves are scary and they can do damage.
Yeah.
But I mean, there's safety things
Yeah.
in protocols and whatever to avoid that. But there's that one, and also the centrifuge. So not the tabletop centrifuges,
The big ones.
but the big ones that are on the floor.
They have like ultra centrifuge, like all these.
Yeah,
ones.
and those are, I don't know. There was one time that I put my samples in, and I thought it was balanced.
Classic undergrad.
No.
Yeah.
Did it not stop?
It was not balanced. I mean, I don't remember. I don't remember because
stop
I was
automatically?
panicked. So I was like, stop.
Depends on like the safety feature. Like sometimes they
I don't
can
know.
stop
They might
automatically
have,
when they
but-
like send something.
As soon as it started kind of shaking, I was like, stop. Yeah, like I was freaking out. So if it was automatically stopping, I wouldn't know because I immediately pressed stop.
Okay, well, it's good that you were there.
You were fast.
Yeah.
Oh yeah, wait for your centrifuges to
That's
get
it.
to the highest speed, or at least halfway
Just
there.
keep your
please.
hand on there.
Yeah.
just
Unless
make
it's an
sure.
hour.
No, to get to the high
Just to
speed.
get to the
yes.
the
Yeah,
speed
for, like,
you
the
want it to
first-
be.
Yeah, wait till it hits the G's and then you go.
Actually, with the autoclave, when I was trained, they said like wait until the autoclave reaches like the temperature and like continues to do like the next step because most of the times, like depending on the model, but the one I was using, they said like when it fails, it's like within the first like step of trying to reach pressure and temperature.
Yeah,
Okay.
same.
How long
Yeah.
does that take?
I don't know, like 5 minutes.
Oh, okay. So you're just like, okay.
Just like yeah.
At least yours is like in the same general room, like
Yeah, the one I have right now. But like before in undergrad, it was like
down the hall
in the basement.
Oh,
my
no!
gosh.
Yeah, and the autoclaves would be like as big as like a deli fridge. And
Oh,
it
yeah.
was the kind that you like slide up and like pull something out.
Oh.
Yeah.
Kind of like a morgue.
Slide
Oh.
up or like the twisty.
Oh,
The
we had
twisty doors are
both.
wild,
The
too.
twisty is kind of scary because they're like, well, make sure it's not too tight, not too loose.
Yeah.
And I'm like,
I'd rather have the little sliding door.
I'm 19. I don't know what that means.
I'm 19, bro. They trust us.
Well, they don't
that's
trust
fair.
us. They gave us safety training and then let us do stuff.
Well, they give everyone safety.
Yeah. I will say, okay, I have another horror story, and this is maybe something
Related?
This is like actually bad though. Like I did a bad thing, and it was not my fault. It's because I was improperly supervised. I think.
Oh.
I would run gels all the time, and I didn't know the light was bad for you. Yeah, and I think that's why I have glasses now, because I would just look into UV light
Oh no.
With your naked eyes.
with my naked
Oh
eyes.
no.
Did it not hurt?
No.
Oh, okay. You were like, I'm invincible.
It didn't have
I can
anything
see
on
colors.
it. Not even like.
It
A
had
cover?
a removable shield.
But you
It,
would
it wasn't
remove the
attached.
shields.
Yeah. I thought it was just to like keep it covered. I know. I told my PI this too. We have a blue light box in our. We don't even have a UV light in our lab
Yeah.
anymore. So I'm like, yeah,
But like, how did you find out that you were doing it not safe?
I took another safety training, and they were like, don't look into the UV, and I was like,
Oh no.
why did no one give me UV glasses? You know what I mean?
Oh no. Not even a. Yeah. No.
You know?
Yeah.
Yeah, because I see some people that have UV glasses and then the shields.
Uh-huh.
Yeah.
And I'm like, I didn't have any of these things. No one gave them to me. Anyways, it's fine.
Oh.
It's fine. I got health insurance now. We good.
No.
What? Glasses are expensive.
They are. I agree.
Okay,
Anyways,
so let's see. So I also couldn't really remember a lot of like silly like mishap and like all these things. But I was in my first lab and I was trying to learn how to save these bacteria in the freezer in order to like go back and use them later. And my grad student mentor said, okay, here's how the system works. Like each box has 81 slots and we're going to start from this number in this box. And here are the tubes I put for you. So on the first one, put 496. And I'm like, okay, cool. And then he's like, all right, I'm going to come back later. You sit here and label tubes. And I'm like, glamorous. And then when he came back, I labeled the entire box 496.
Oh no.
And he was like, this is wrong. And I'm like, oh, I have to go to class now. Sorry. And then he had to fix all my mistakes.
Oh no! Well,
But
he
it's
should have
fine.
instructed you correctly.
He should have said,
clear instructions.
yeah, 496, 497. No, he was like, start from 496. I'll be
Should
back.
Start from 496 and then label them in an ascending order. It's the classic make a PBJ with only the instructions on the paper. He didn't give you the right instructions.
That's true.
Wait, there's an instruction with making PB&J.
There's like a you've never done that. There's like a
We don't eat that in Thailand, but yes, do tell us.
or here or like any like team building thing. They'll make you share a
Oh.
paper
Oh.
and like write how you make a PB&J and then have the other person follow exactly what you wrote. And
Is
then
this
you
like a
end up-
scientific method paper?
Yeah, I think so.
Oh
or
my
something
god,
like
I
that.
love that.
That's fun. We love
I've
team
never done
building.
that.
We
Makes
love
sense.
team building.
But yeah. So
Yeah.
that was not a fun one. But it's okay because I wasn't fired on the spot. And then I kept working with them again
No.
for like 3 years
It's fine.
until I graduate.
You
and
know
we're still good friends.
I think like when you have undergrads in the lab, you just expect them to mess up. Like, just this is all learning for them, and a lot of learning is making mistakes. So yeah,
What?
I remembered a horror story,
Oh,
but it's not something
should I
I
like to
did.
share?
It's okay.
It's okay, you can share.
Okay.
Exciting.
So the way that I was in a microbiome lab in undergrad, and we did a lot of 16S sequencing. But when we went to sequence the samples, we would send it to the core or whatever at UCLA. And what you do is you do this whole process before you send it to sequencing. You like extract the DNA, you amplify it, you purify it. And then at the end, you pull all your samples because they're barcoded with a unique barcode that'll allow you to read all the samples. But you pull them all in one tube. And I remember I think it was 1/4 and it's like finals week. And I had pulled all my samples and I sent it off. And then I get a email or a text from my grad mentor. He's like, they lost your samples. And I was like, what do you mean you lost my tubes? I was so mad, bro.
How did they lose
I
your
was
samples?
freaking out. I was studying for finals. I am under the impression that all my samples are gone, like literally years of work.
Yeah.
It ended up being fine. They found them, but
Okay.
Jesus, like if you think I'm stressed now, imagine me at that moment.
Oh no.
I have like a Snapchat memory that comes up every single time that happens, and now I can laugh about it.
Yeah. At the time, it was not
But
fun.
at the time, it was not fun. So that's a horror story.
Oof.
Yeah, that's rough. That's rough.
Do you have something right now? Do you have another story?
Oh, I have plenty. I like when you're like, I can't think of anything. I'm like, I literally remember everything I've ever done wrong in lab ever. Like I it's just so I won't make the mistake twice, you know? You just gotta
I feel like I have selective memory.
Yeah.
That's-
I know. I wish I
that's
could
my healing
forget.
process.
I
Yeah,
wish I could
same.
forget.
Yeah.
Yeah, I did an internship and we were doing flow cytometry, which requires single cells. And we would take the tissue and then do a bunch of stuff to it to get it to be a single cell. And some of the steps involve a centrifuge. And the centrifuge that I was using was a little old.
Mm.
So we lost a lot of the cells because they didn't like pellet down. So when you like pour off the liquid, a lot of the cells were still in the liquid. And then one time I accidentally used like the wrong centrifuge for 2 of the samples that we were doing flow on, and the right centrifuge for the other 2 samples. So I literally got a comparison of how many cells I lost, and it was like over 4 million cells per tissue.
Oh no.
I'm like, yeah, that one was rough. And then
Oof.
yeah, and this is why I did not have a good time. I loved this internship. I was like when I was in Boston, and I did not. I was like unhappy in Boston. It was like not the place for me. So I was
Wow. First thing I've heard of.
just, what?
No, she said this
Nothing.
before.
I'm kidding.
Oh,
First time you've heard someone say
that they don't like Boston.
Yes.
Yeah,
Yes.
I'm
Yes.
the only one that hates Boston.
Just kidding. I'm from Boston.
Okay. Yeah, I don't know. Everyone's
My
like,
next
"I
story
love
is
Boston."
about the
Okay.
very same institution.
Oh,
But
my.
You continue.
Yeah. So, one time I think we took, it must have been like 16, maybe 20 mice, like samples from 20 mice, and I used the wrong centrifuge, and I didn't realize until later, and I'm like, I lost like, I don't know, most of whatever. Like it
Oh,
was like,
no.
so, it was so bad. And I remember going home. It was like 9 p.m. And I remember going home and just like crying because I'm like, bro, how did I make the same mistake twice? And that's why I remember all my
don't
mistakes.
Cry. Never
it's okay.
cry over science.
I know, but like
Sometimes it just really
you're
gets
like,
you.
like it was like a crying out of frustration of like,
Yeah.
oh, I know better, and like I can't believe this happened,
Yeah.
and It's like an internship at like this fancy institution. You want to make a good impression. And it's like the impression I gave them is that I used the wrong centrifuge.
Oh, that's okay. Maybe they should not.
What if
be
they
using
forget?
the centrifuge.
Why don't they just unplug the centrifuge? Put a different centrifuge
I
in the
know,
cell
bro.
culture room.
Exactly. Bro, the day that I stop crying out of frustration, it's over for y'all.
Listen, I wish.
There was this one time. Okay, I don't know. I know Asama might understand this feeling, but you get through like a thing, like you're doing a bunch of bacterial culture, and you want to then you've done this whole process, and then you finally want to plate your cultures.
Yeah.
Have you ever forgot to dry your plate?
Oh,
And
no.
it's like a time-sensitive thing, and you're like, bro, do you work with bacteria
Like
at all?
before?
No.
Not anymore.
Like dry it before you plate it?
Yeah. Or do you not
because-
dry your plates?
Well,
Because it's condensation.
depends. Yeah.
Like it's been in the
Flip 'em
cold
upside
room.
down.
Yeah. How do you dry
Onto
your
the
plates?
table?
Yeah, but you don't stack them in the fridge upside down.
Yes.
It's still, but they're in the cold room for forever. So,
so when they
they're
come
still
out, they
yeah.
sweat a little.
Oh,
Yeah.
Yeah.
How do you usually dry your plates?
I flip it upside down with the lid, and then I just like,
Like the lid on the bottom.
the lid on the bottom,
And then you put it at like room temperature?
Yeah, but I open it a little.
Oh.
I
can you just like
don't put
flame
it in room temp. I put it in the hot in any
warm plate?
Incubator.
warm room. Yeah.
Yeah.
Yeah,
No,
I've
because
done that,
unless
but some
it's
people
gonna
were
just
like,
sit
you
there.
can do that, but in the BSC, because like all the airflow, like the biosafety cabinet, like the hood,
Yeah.
because all the airflow like helps the sweat wicking situation.
Wow.
Yeah.
But yeah, so that's. I
But
remember,
yeah, often.
I remember one time I did that, and then because of that, everything got pushed back.
No.
And I didn't leave lab until like 11 at night. And it was like one of the last few days that I was actually also in Boston.
Yeah.
So I was like, bro, I want to go home.
Yeah.
Wow.
Yeah, because I had to do cereal dilations.
Did you like Boston?
I did.
Did you?
I
like
liked
Boston?
it enough to go again. But
Like on purpose.
I feel like I like it enough. Like,
yeah.
like it enough.
Yeah.
Yeah.
I mean, I got my education from there, got my biotech training from there,
Yeah,
met
yeah,
my
yeah,
partner
yeah.
there, my family's there,
Yeah.
my friends are there.
Yeah, it's different.
Yeah.
I think Boston.
I think
Good
it's
enough
different.
things
bit more
going for
home.
me.
Yeah.
Yeah.
In fact,
I would go back.
does this thing say Boston? It says
Don't
Boston.
I don't know if I will.
I love it that much.
Not
I
even
think
a
I was
visit?
scarred.
Mm.
No, I'll go, and like the dance
feel
scene
like-
at Boston was so great. Like
That's something I never got to.
I'm sorry,
Sorry.
for your loss.
Yeah, I was a child.
That was like the best.
We called
dancing.
it being in daycare because everyone else was of age.
Daycare
Yeah.
expensive. Was your daycare free?
Yes. Well, I was paying for my room to live in Boston. those times.
They
Well, I
made you
didn't pay, but
Okay.
my first summer they paid directly to the people to have
Yeah.
a room, and then the next summer they give you an increased stipend so that you
You can pay for it,
Yeah.
okay.
So I did not want to live in the dorms that they had us housed
Yeah.
in because I was scarred by the mice.
Oh no, not the mice.
And so the second summer I went back, I just subleased. I was like,
yeah,
did not see a mouse once in that apartment.
I love that
so-
for you.
Okay. Yeah. So in preparation for this episode,
Oh,
I had
my.
asked people like, hey, have you ever heard of any spooky or horror stories? And they were like, no, luckily, ha ha ha. But have you heard of that 2009 coffee poisoning event at
What?
a
Excuse
big
me, what?
institution at Harvard, which, by the way, is on ABC News? So I
the
will
what? The
maybe
A coffee poisoning event in 2009?
Take coffee away from me.
add a big fancy medical institution in Boston.
On Fenway, by any chance?
Yeah, in the Longwood area.
Okay.
Yeah. So maybe
Oh my.
maybe some of you know about this already, but
Dang.
in brief, people were like, so there's this sort of like communal like coffee or like lunchroom on
Mhm.
this floor on the like pathology like
Mhm.
unit. And usually someone makes coffee and then you're like, okay, yeah, there's a pot of coffee. Let me go like
Mhm.
get some. And then so people did that and put in, their usual like sugar and milk or whatever. And then nothing happened yet
Mm-hmm.
until this one person, Mateo.
Oh, we're
usually-
naming names.
I mean, he's named in the ABC article.
He's on
Uh,
ABC News.
He's on ABC News.
Drinks his coffee black. And then he's like, something's going on. Like, this coffee tastes bad, but like worse than usual.
Oh.
And they, like he drank his coffee and then he's like, you know what? Maybe this is just a bad batch. I'm I'm not gonna continue drinking it. But other people that had like a cup already started to like sweat, started fainting, they were dizzy, and people were like, "This is unusual."
Yes.
So long story short, they did toxicology study and
Of the
found
coffee.
sodium azide in that pot of coffee, which is a chemical that's usually like used to poison or like kill bacteria.
Did they find how it got in there?
To kill bacteria. So it must have been
Like a cleaning.
like a
thing?
cleaning.
But it's like a very to kill bacteria in your experiment. So it's like very specific, like
and
potent
that's why
activity.
you take your gloves off.
But so my friend who told me about this story
Mm-hmm.
was training in this program during like her PhD at this institution, and she said that someone did that on purpose
And they
to
didn't catch who it
spite
was.
the people.
That sounds very illegal. You could have killed somebody.
It's very illegal. That's why ABC News is like, oh, this is a good news story.
Oh, no.
But in
Oh, wait,
case you
okay,
didn't,
what is the end of the article? What did they figure out
I
who it
didn't have
was?
enough time. I was
I'm sorry.
listening to this.
Can we look? Can we look?
Okay. Blah, blah, blah. Concentration of sodium azide. Every lab has it. Very common. This guy's like, oh, I drink my coffee black because I'm Italian and I knew something was up.
Love that.
Blah, blah, blah. 2 people fainted. 6 people got hurt in one way or another.
The 8th floor coffee machine.
The spokesman for this medical school said,
We are now going to the Starbucks downstairs.
we're not connected to this.
I'm dead.
So funny.
Well, I'm glad the Chemex is in my office.
Okay, but that's the thing. Like our floor, we have a coffee pot for the floor. I don't drink coffee, so this is never going to happen to me. But
Yeah.
there's a little nerve wracking that we just
Yeah.
be drinking things.
Yeah.
Yeah.
Oh no.
Yeah.
Well, communal areas are now ruined for me.
Yeah. Thanks, Asama.
Well, it was for me before because I'm a germaphobe and because of the pandemic.
Yeah.
But yeah, watch out.
Watch
Never take a
out.
free donut
No, whoa.
or
Whoa,
anything.
This is too much
This
for
is too
me.
much.
Don't
Yeah.
ruin free
This is too
food
much
for
for
me.
me.
Sorry.
It's about to be Halloween. I want to eat all the free candy.
This is
Free
too
candy from
spooky.
where?
What do you mean? I don't
From
know. I
where?
like parties and stuff. They have the candy.
You're going to a party?
Oh, not I. I'm not going to a party. We're in a pandemic.
Anyway, if you do go to a party, make sure everyone's vaccinated and be safe. Thank you. Bye.
Safety.
Yeah. Okay. So let me. Does anyone else have any other stories?
Oh, can I share stories about poop?
Oh yeah.
Sure.
It's kind of like silly / a bit like
Sure.
scary at times. Okay,
Okay.
so before, similar to Lennice, I also had my undergrad / pre-grad school background in microbiome, and one of the places to really like dive into this sort of environment is in the gut. And yes, Lennice said yes. But Lennice did fish gut, which
I
is
did
like
do fish
really
gut.
cool. I did
Yeah.
human gut, which is like cool, but sometimes you have like people line up outside your lab waiting to poop for you.
Oh no.
So
Do you stand outside the door and be like, "Are you
Actually,
done? Did you have
yeah.
a good one?"
yeah. And then, well, unfortunately, I was not involved in the like, hi, here's your poop bucket, like part of things because I was
Did you
was
wanna be?
doing science stuff. Kind of. Like, it's super fun.
So you're a germaphobe, but you want to handle people's poop?
Not like with my bare hands.
Oh.
Gross.
But I want to talk to them about their poop because I think psycho. Anyways, so we basically were working on a poop project, and we would, as one does, like go to conferences about like microbiome stuff, blah blah blah, exchange stories.
Mm-hmm.
And one of them was, this is again at the same unnamed institution as the poison coffee place.
Oh no. Well, I think we kind of.
What's done is done.
But someone told me
Allegedly?
that someone from also a microbiome lab was transporting a poop sample from a donor to like their lab,
Mhm.
and they were like so frustrated that they like crack open that container, smear the poop all over the elevator, and left.
Allegedly.
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 Oh no.
But it's a
Bro.
known story in this circle at this place during that era.
Yeah, but is it like, did it really
Gross.
happen, or is it just like a thing people tell? Like,
I can try to
tooth fairy.
find an ABC News story
Okay,
for you.
Need
please.
facts.
That's so
gross.
funny.
Okay, well, not as bad. but still related to poop, since I also did microbiome research. I love conferences. I love talking to people to an extent because I'm shy. But at conferences, you sit at the dinner table, right? Like with a bunch of people you usually don't know, maybe a couple friends.
Yeah.
And you're like eating dinner or like dessert. I love my research, but I hated having to be like,
I do poop.
I do poop. Yeah, like I research poop. Like that's what I have to talk about. So that was like always like at conferences. So not a horror story, but kind of like
we always talk about poop at my company, the poop company.
Yeah, I feel like at that point you're like,
and like they were so into it that you would wear like a mascot, a poop emoji costume mascot like once a month, and hold up a golden plunger and like announce like, oh, giving a shout out to this person, that person,
that
and like. Well, hopefully everyone in the company pooped that much. But it's like,
check your bowel movements.
It's like announcements. And I thought it was so cute. And the best part I thought was that if you're like employee of the month, you're called a Bristol 4. Do you know what that means?
Oh, isn't that the scale of like how good your poop is?
The scale of how, it's the scale of how good and perfect composed your poop is.
I hope no one's eating dinner. I'm so sorry. Yes, Amanda? Comments? Concerns?
I am concerned.
And I think they get like a poop sequencing like kit as a prize, so they can know exactly.
Sequence their microbiome.
Yeah. Which is fun.
That's not their entire microbiome.
Just whatever.
You probably lose a lot of.
Yeah.
Yeah.
Anyways, that was enough conversation about poop, I think.
I'm just getting started. Sorry. Continue.
No, I have nothing to say. I just would like to move on.
Okay, I know me and Asama have essentially just taken over the conversation.
You're totally fine.
But like,
I mean, I never do usually.
Asama, isn't there like a ghost in your building?
Oh my god, there's a ghost in your building?
Oh my gosh, yes! Okay, so when I was rotating the older grad students who have been there for like a couple years said, hey, like, do you work nights by any chance? Like, make sure that someone's there with you, just so you know there's like an accountability partner, which is a safe thing to do because I was new.
Yeah.
So I was like, yeah, okay, thanks so much, blah, blah, blah. And as time goes on, people were like, oh, have you heard of the ghost? And I'm like, what ghost? Who? What do they look like?
What ghost?
And apparently one of the labs on our floor, the mannequin lab, would sometimes have like unusual like time points, like at the 12-hour or 13-hour. So maybe they have to come in at like 9 p.m. or 10 p.m. to like take care of their science. And some people have reported seeing this like blonde Caucasian man individual in a lab coat.
What?
Just like,
isn't this a new building too?
It is a new building. Who died in this building?
Yeah, but every building has some sort of...
Yeah, we built on the land of other people.
Yeah.
Yes.
Yes.
That's facts.
But I was like, oh, maybe they're just like being silly because they're staring at the microscope for a long time.
Yeah.
And maybe they're just seeing things.
Yeah.
So I just like didn't pay attention too much about that. But recently I had been doing this time point situation as well, where it's like a 13-hour. So I would go in at like maybe 7 or 8 p.m. And I'm in my teacher culture room, which has a swing door that has to be activated to like open. It's not just like out as part of the main lab. And usually I can tell if someone is coming to use like equipment or like some other things in the room because the door will click when it's like senses someone or something there.
Yeah.
So I was there at night and I heard the door clicking and I was like, oh, just come in. Like, I'm just here.
No, don't
And then no one said anything because like during COVID, like we wanted people to feel safe, like have properly
like
distance space. Yeah. So we were like, oh, if you don't feel comfortable with someone being in that room with you, like if you say no, we won't bother you.
Yeah.
So maybe that person didn't hear me. So I was like, okay, whatever. They'll just wait. And then when I'm done, I got out and I'm like, okay.
Hello?
Are you there? And no one was there. I checked the calendar. No one signed up.
Oh, no.
I walked through all the rooms, like the back rooms, the office. No one was there. So I'm like, what motion was it sensing?
I don't like that.
I don't like that at all.
So it's possible that it's faulty, but...
Does this ever happen in the daytime?
No.
Oh, bitch.
And no one else has noticed it. Or when it happens in the daytime, when I hear the click, I'm like, oh, someone's at the door. I come out, someone's there. And they're like, oh, yeah, we were just waiting to see if you would feel safe for us to come in. So I don't know. I think I have a friend, and it's kind of nice.
We're befriending Casper.
Can you help me do my serial dilution? That would be great.
If you ask me and I don't have any, if I'm not in the middle of anything, I will go with you to
so you can scream with me.
I'm scared, bro.
Okay, you got to tell the ghost to leave you alone. You can't say, come in, ghosty, be my friend.
I mean, I'm gonna be here for the next 5 years. I want to be on good terms with the ghost. Yeah. It's fine.
That's one way to do it.
My bags.
That's one way to do it. Could not be me. Could not be me.
Yeah. So
okay.
Yep.
All right. Well, I think that's a great story.
That was great. A great story to end on.
Thank you for sharing, all of you, with us all, the stories, ghost related or not. And I'm happy that we were able to tell you guys some spooky stories pre-Halloween. Or right when is this coming out?
The 29th.
The 29th. So yeah. Pre-Halloween. Great.
Mm-hmm.
Okay, so now let's take a quick break before we say goodbye.
Sainted Factory is a Black-owned skincare brand that prides itself in providing natural, homemade products.
If you are a skincare buff and love to have the taste of luxury at home on a university budget, these products suit those needs.
They have 3 scented exfoliating scrubs that are catered to help different skin conditions.
As you head into another school year, having a scrub that not only nourishes your body but also helps you relax after long days are a must.
Yeah.
And now you can use our code TriplicatesPod10 for 10% off your order.
Yes, I love that.
And you can get them on saintedfactory.com. They're really cute. I use them a lot. Treat yourself.
Okay, welcome back, everyone. Before we go, we wanted to ask, what are you going to be for Halloween?
Oh, I go first?
Yeah.
I guess so.
Well, I have a group costume with Amanda.
Oh, and would it a group if it's just 2 of us?
Would have been Lennice too, but she's occupied.
I'll be out of town.
It's fine.
Yeah.
Yeah.
Do you want to tell them?
Yeah, we're gonna be the Winx fairies.
Yeah.
The Winx Club fairies.
I'm gonna be Musa.
Yes, I'm gonna be Aisha, who is also Layla, depending on which version of the Winx Club you watched. But
Cool. I already got my items.
Yes, I can send you. I
want to see. I've essentially seen your outfit, but I want to see yours.
Okay, I'm trying to figure out if I should go all out with the shoes.
Yes,
but then it doesn't get cold here, so I'm like, well, if I just buy
doesn't get cold here.
something.
I'm cold. All
I mean,
the time.
I'm wearing the most.
Yeah, maybe.
Booty.
I don't know.
We can discuss after the show.
Maybe we can go.
Yeah,
shopping or something.
But
I've been looking at red boots.
Why are
I think
you
they're
down when you're not here?
I will go shopping with her before the event.
The event that's this weekend?
Well, when is she gonna go? Stop yelling!
I'm sorry.
Okay, but the thing is, I feel like Flora, you already have clothes for Flora, so maybe we could just dress up for something.
Yeah.
Okay.
We'll
see. Okay.
There's
anyways, Amanda.
Yeah, I'm gonna be Aisha from the Winx Club, and then I might also be a demon slayer, who's also a demon who slays other demons.
Wonderful.
Yeah.
Oh my god.
I got to fit.
The guy from Charmed.
Who?
He's a demon, and he also slays other demons.
Oh.
No. Charmed?
I didn't watch Charmed. I was giving
Not the new one,
Mazikeen
the
from
old one.
Lucifer.
Oh, yeah.
That was what I was giving.
Wait, so can people just show up as demons or demon slayers? Or
you can
does
show
everyone
up
have
as
to be
whatever you
It's
want.
Eldritch Aberrant, whatever that means.
I didn't know what that meant.
I didn't
Have
know
you
what it
watched
meant either.
Sabrina, the Chronicles of, or what is it called?
Is
I don't know. The Sabrina, the Netflix one, not the old one.
Yeah.
Anyways,
it's fine.
And I will not be the Winx
No, that's okay.
fairy, although I really would have loved to be, but alas, I will be out of town. But I'll be Sabrina from Sabrina Teenage Witch or whatever. The new one is.
Whatever.
I forget what it's
It is
called.
the Chronicles of Sabrina.
It might be. Oh, the Chilling
The
Adventures of Sabrina.
Yeah, I was like, there's a C in there.
So I had a bunch of things that kind of match her aesthetic.
That's
So
Ooh.
I'm excited.
cute.
I'm just not
Love
blonde.
that.
You don't have to
But
be.
here we are.
It's okay.
Okie dokie.
All
So,
right. Are we ready to round out the show?
Yeah.
You guys want to run some observations? Do some statistics?
I mean, of course, it makes me a good scientist.
Yeah.
So the question is, how many PhD students in our study would rather handwrite notes from a PowerPoint or make PowerPoints from handwritten notes?
Reading somebody else's handwriting?
No, thank you.
Writing my own? I feel like I trained to write notes from PowerPoints, like handwrite notes from PowerPoints.
Yeah.
I feel like I could do that.
Well, yeah, that's like watching a lecture. No.
Yeah. This is in the context of like lectures, right? And not
I
like
guess so.
giving a talk.
No, I don't think you have to give a talk.
Okay.
I've definitely had slides written out what I want to say. They weren't my slides, and then given the presentation. So I'd rather do that.
I'm sorry, I don't understand.
Like it was
Say
someone
again.
else's slides. I
Yeah.
hand wrote notes on how to give the presentation based on the slides.
Oh, and you're
and then
like,
gave the
you're like,
presentation.
yeah, okay.
Yeah.
I gave it.
Oh yeah,
because they
yeah.
weren't my slides.
Yes.
Yeah.
I see.
Oh, I was
see.
there.
So you
So I'll do
hand
that
wrote
one.
notes on the slides and gave.
Yeah,
Okay. Yeah. Yeah, I think I'm gonna handwrite notes.
I would say depending on the purpose. Like if I'm in a lecture, I would rather be the one writing my own shorthand or like trying to conceptualize stuff. But if I'm giving a talk, I think I prefer making slides.
Yeah.
Because I can draw other pictures, especially with like biological processes. Pictures are so powerful.
Yeah, that's true.
Yeah.
Okay. Lennice?
So I don't.
66?
You already
I
answered.
guess. Which way? What did you say?
I said handwriting.
Okay, so 66% of our PhD students would rather handwrite notes from PowerPoint, while 33% would rather make a PowerPoint from handwritten notes. And that is fair.
It's
Is
fine.
that statistically significant? Sorry.
Well, we have an N of 3, so really anything that we do would barely be statistically significant. And some would argue that that's wrong.
Anyways.
Anywho,
I mean, we're not really independent samples.
That's
we're
fair.
pretty much linked.
we have 2
Yeah.
brain cells
That's
That's
true.
that we share.
Wait, did you see someone like entered in their department Halloween pumpkin carving competition with like 2 brain cells because they're like neuroscientists, and also like
Oh, that's
Like, we got 2 brain
it's
cells.
hilarious. Yeah.
That's fun.
Okay, so Asama?
Cool. So if there's any other topic you would like to hear from us, just let us know.
You can find us on Twitter, Instagram, and on TikTok at
Ooh.
TriplicatesPod. And you can email us at triplicatespod@gmail.com. If you like us, support us on patreon.com/triplicatespod. And with that,
Obrigada e ciao.
gracias y adios.
Kapun ka. Bye. Alrighty. We should have a dance party.